Hi, Aaron. Hi.
Hi, Norbert. How are you?
It's okay. How are you? Good to see you.
Good to see you.
Thank you. Shall I try to share my screen? Maybe.
Yes, please. Yeah. Yes, please.
If I have a. Yeah. Can you see it?
Yeah. Do you want to do the slideshow?
Yeah, absolutely.
Yeah, looks good.
It looks good.
Yeah,
okay.
Thanks so much for doing this.
Oh, that's my pleasure. Thank you. And can you see my pointer as well?
Yes, perfect.
Okay. Oh, offset then. Thank you. Then I will stop sharing. I think Nash will be the first presenter. Am I right? Oh, great.
Right. Are you in Indiana now?
Yes, in Indiana. Yeah.
Got it. Okay.
Yeah, absolutely.
Good. I came by a couple years ago. Huijian invited me. I think it was before you got there.
Yeah. Actually, I just came last year. So I was recruited more than 2 years ago, but because of the pandemic, it was very hard to move the whole family overseas. So, but finally, we just came here. That's really great. Yeah.
Great.
Okay. Yeah.
Yeah, that's a classic American college campus town.
Absolutely. I enjoy it a lot. We just had a visitor actually this last week from Stanford, from Ivan Scholtil's lab.
Oh, great. Okay.
Yeah. I think you know him. He made a presentation maybe a year ago here at the Neuro Zoom. Ivan
Yeah. He's amazing. He's from Hungary too, right?
Yes, absolutely. Yeah.
So okay, yeah.
He's Hungarian.
Yeah, he's really amazing.
Yeah, and one of his colleagues was here at the IUB and give a lecture.
Oh, great.
Yeah.
Yeah, that's wonderful. How's the weather? Is it too cold or it's not too bad in Bloomington?
It's okay. I like snow and freezing as well during the winter time. So first I used to it. Plus, it's Hungary nowadays. There is not a real winter because of the warming. So you're just facing pretty warm weather. But here is pretty okay. I like it. Skating, etc. Yeah, that's good.
Good.
Okay, I will mute myself and then we'll come.
Thanks again.
Okay.
Yeah.
Aaron.
Hey, it's long. How's it going?
Yeah, pretty good. Are you there yet? No.
Nash is here. Norbert tested his slides.
Sure, great. Hey, Nash.
Hey, Nash.
So the ARS meeting will be in the campus of the Shanghai Tech, right? I believe
Yeah,
yeah,
right. It's Institute for Advanced Technology.
Yeah, and it's
I love.
great.
Yeah, in person. So I love the pace of China. It literally took me one afternoon to organize this 2-day meeting, whereas I'm organizing meetings in America. It takes like 2 years.
We organize all the meetings through WeChat. You know, you get all the confirmation, all the talk titles.
Yeah, I haven't sent a single email about the meeting. Everything.
Yeah.
Hey,
hi, good morning.
Nash.
Oh, hi, Aaron. Hi, Tulum. Good morning.
Yeah, works great. Works great.
Sorry to disrupt your conversation. Can you see my slides now?
Yeah, sure.
Do the slideshow. Yeah.
And if I make them presentation, slideshow, how is it now? Yes.
Just a second. It was slideshow. It was slideshow a moment ago, but it still wasn't now.
Shared application,
Click.
please. This window.
Yeah, just a second. Yeah, I know. Well, I saw you clicked, but haven't.
Did you click on it?
One participant can share at a time. Multiple participants, should I use that?
Huh?
I don't know. Huh?
Try it again. It was slideshow before. Yeah.
Can you see that?
No.
Okay. Let me try again.
Sure.
Okay.
Okay, so we have this part, minimize this screen, and then we take the PPT out and Zoom meeting, unmute videos there, share screen, increment for video clip, whiteboard, share. How is it?
Click on the slideshow.
He's already telling me that participants can see your screen now.
Just click on the slide. Yeah, click on that thing.
Yeah, I'm clicking now.
Yeah. It should work.
Now it's slideshow.
Weird. No, we
don't see it.
Yeah.
You don't see that?
Yeah, I saw it clicked.
No.
It was a moment ago, but not right now.
Mm-hmm.
Maybe quit your PowerPoint and reopen it.
Okay.
Yeah.
Let's just close. Screen sharing has stopped as the shared window is closed. Okay. Let's reopen again. Is my computer loaded actually in this case? I'm wondering whether. All right, Neuro Zoom folder.
Try it again.
Go. PPT. You guys still can hear me, right?
Yeah.
Oh, yeah. Oh, this Neuro Zoom is very dangerous.
I know how it works in Fudan. Did you work on this show at home or in campus?
No, I'm working from campus.
Okay.
And it works from here, as long as you are joining a meeting.
Sure.
Okay.
Share your screen.
Yes, 1 second, just 1 second. Now we go with share. You see that?
Okay, so go, yeah. So go here, view, and then do slideshow. Oh, that's not it. Sorry. Go. Sorry.
No, that's not what it is.
Go here. Slideshow.
Slideshow here. Slideshow.
Yeah.
Yeah.
And just go from the beginning.
Sure.
Yes. Do it. Yeah.
Go from here, right?
Yeah. Sure.
Yeah. Click that.
Go ahead.
Now, how is it?
Just a second.
What does it show on your? What does it show on your screen? Nash, what does it show on your screen?
He's asking me to please move this window away from the shared application.
What does it show on your screen?
Nothing. On my screen, I see a full slideshow.
That's weird.
And your pictures.
Do you have multiple? Okay. Do you have multiple? Do you have multiple displays?
Participants can share simultaneously. Should I do that?
Yeah. Sure.
Yeah, maybe. Yeah.
Now, how is it? Participants.
No.
Am I having something locked on the security? Allow participants to share screen, chat, rename. It's so correct. Everything is right. It's the default. And I used it before. Why today it's like this?
This is the main reason we ask people to come early to test things out.
You know, I have used Neuro Zoom before that. And I give a talk before in a meeting, was in Europe. And
well, that doesn't matter. What matters is it's not working now.
Now, exactly. That's what matters now. So sharing options, one participant can share, not only host or participants who can share. I don't think this helps, like who can share single participant or more. So if I don't make full slide show you guys. Oh God.
All right, we'll try something else. Share your screen. Do you have movies?
Yes. But it doesn't matter. Now, how about this one?
That's good.
Yeah, exactly. Good. Okay, so you need to make it a full slideshow on your screen first and then share the screen.
Yeah, go ahead.
Okay, so you see my screen now, right?
Yeah.
Right, so if I start flipping up here.
Go to the next slide.
I'm going next. No. Video, does the video, is the video going fine?
Yeah, all right,
great.
We can, okay. Excellent. So go back to the first slide.
Forgive me, because I have many videos. I just want to make sure that it's going all right. Okay, so the key for me, at least for my computer, I should make a full slideshow first and then share the screen.
Whatever you have to do, but go to your first slide.
Okay. I think it's all right now. It's safe. Okay. Yes.
Okay, now stop sharing.
Okay.
You can start it.
Okay, yeah, now we're a little bit late, so we'll get started.
Zoe, good morning. How you doing? It's great to see you.
How are you? Great to see you.
It's so great to see you.
It's great. It has been such a long time.
Yeah, really long time.
Yeah,
I'm glad that you survived this COVID thing. Oh.
Yeah. So hopefully next year we will make it back to China.
Yes.
Yeah, so we really want to visit.
I think we will make it. We will make it at the end. Don't worry about that. Okay, sorry for the disruption, guys. Go ahead.
Yeah, all right. You guys can chit chat a little bit later. We'll get started now. Welcome everyone to Neuro Zoom. Thanks for coming. We have two awesome talks today about exciting new neural circuit studies. And before we get started, advertisement for next week. We have Julia Kaltschmidt from Stanford and Honghui Zhang from the Allen Institute. Those talks aren't to be missed, and so please tune in then. And please let Zilong and and I know that if you're interested in presenting your research at NeuroZoom, we have slots available in the spring. Okay, so it's my pleasure to introduce the first speaker today, Nashat Abumaria, known in China as Nada, or known elsewhere as Nash. So Nash received his undergraduate degree at the University of Jordan. Then he did his PhD in Germany in neuroscience at Göttingen University, where he studied the role of stress in the induction of gene programs in response to stress. He then did a brief postdoc at the German Primate Center, and then moved to China, where he did a postdoctoral fellowship with Guosong Liu at Tsinghua University in Beijing. And here he had a really neat discovery about elevating brain magnesium levels and the effects on enhancing synaptic plasticity. He rose through the ranks at Tsinghua, he was promoted to associate professor, and then moved to Fudan University in Shanghai, where he's a tenured professor in the Institute of Brain Science and Department of Laboratory Animal Science in the School of Medicine. His lab is unique in that it uses hardcore behavioral studies in animals, which you'll see today, combined with molecular mechanisms. He had a really interesting finding in his own lab where he studied this protein TRPM7, which had been studied by others in a role in pathology and in neuronal damage, but Nash discovered an unexpected role for it during normal physiology and found that it wasn't actually the channel function of the protein that was mediating this, but rather a kinase domain. So this is an example of how Nash approaches a scientific question from sort of a different way than others in the field have, and he doesn't always accept conventional wisdom, and these approaches have led to key insights. Another note about Nash, he always shows up to things. This is important. This is an underappreciated behavior in science. Like, he shows up, he's always at meetings, you always see Nash there, he actively participates, he's a force for good in science. Showing up is important, and on that score, nobody does it better than him. So he has a really interesting talk coming right now where he's going to present something that you'll think is one thing, but he's going to try to convince us that it's a different thing. And go for it, Nash.
Okay. Thank you, Aaron, for this beautiful and wonderful introduction. And I also would like to say that Aaron also attends meetings, and he brings life to our life here in China, creating lots of meetings, interactions between the scientists. So thank you so much for making the life more interesting than it was before you came here. Many thanks to Aaron and Zolong for arranging this beautiful meeting and for giving me this opportunity. Today, I will be sharing with you a story that has been evolving in my lab over the past 5 years. We will be talking about a neural circuit that regulates give-up like behavior. Can you see my screen now and you hear me? Everything's all right, okay? All right, so perfect. Let's take off. So give-up is a very critical adaptive decision-making process. When individuals face challenges, it's very important that they face it with actions. They try to overcome the challenge to avoid being ineffective or inefficient. But at the same time, they also have to be sensitive enough to detect potential futility so they can change the behavior, switch the behavior, or change their strategy, or even give up to move on and do something more productive if the task turn out to be futile. Some studies in human, particularly imaging studies in human, do provide us with some insights on the cortical brain regions that might be involved in signaling controllability, failure, and quitting behavior. Nevertheless, the exact mechanisms underlying this adaptive decision-making by the brain, basically to switch behavior or quit or even give up, remain unknown. There is a major challenge in this area of research, that is, we don't have animal models that could enable us to elucidate the neurobiological substrates that might be related to this kind of behavior. So far, there is one beautiful and elegant study that has been published recently, showing the inhibition of unsuccessful behavior in zebrafish when futility is detected. The authors here have shown in a very beautiful and elegant way how norepinephrine with the neuronal and non-neuronal components work together to detect uncontrollability and to activate mechanisms to inhibit unsuccessful swimming behavior in zebrafish. So if we look carefully in the literature, we will find that there is an old hypothesis or theory in cognitive psychology made by Johannes Kuhl, the German psychologist. In 1981, Kuhl has said that when human subjects are facing a prolonged uncontrollable experience, they immediately act by taking actions. And he described this phase by saying because they are in a action cognitive state at this moment, so they start taking action, they try to overcome the challenge. However, if this uncontrollability or this failure is maintained for a long time, then the subjects will start to transition to another cognitive state. He called it actually a state cognitive state, during which they will take no actions, they will not do anything to overcome this challenge. So in our lab here, we thought whether or not we will be able to create some prolonged uncontrollable experiences to animals and observe their behavior to see whether or not we might be able to see such a behavioral transition from action to no action in these animals. Of course, many of the people who know me and where I come from, they know that after that, we need to do some validation for the models that we are trying to establish. And we want to use these models, try to elucidate the neurobiological substrates like neuroanatomical and neurochemical substrates underlying this behavior, hoping at the end to provide you with some insights on the following question, like how the mammalian brain switches behavior when facing prolonged uncontrollability. So here we created two behavioral paradigms. In the first paradigm, we exposed the animals to inescapable electrical foot shock, and we monitored their behavior during the exposure to the shock, not before, not after, during the electrical shock. And what we noticed here, we noticed that the animals started to react at the beginning during early trials, started to react in a fierce manner. They are biting the ground, they're trying to jump to escape from the place, they are running in a circular way in a very fierce manner. And then later on, when this whole thing is prolonged, and once the animal realized that there is no escape or no control of this situation, the animal started to exhibit complete immobility during exposure to the electrical shock. So what you see here, you see an animal that is doing nothing while it's being now exposed to electrical foot shock. We classified these two kinds of contrasting behaviors as action and no action. Now, when we plotted these behaviors, we have noticed that over 3 days of exposure of a prolonged experience, uncontrollable experience, we can clearly see a gradual transition from a behavioral pattern that was dominated by action on day 1 to a behavioral pattern that is dominated by no action on day 3. Now, was this behavioral transition because of pain desensitization? The answer was no, using 3 different tests. Was this muscle fatigue? The answer was no, according to Rotor challenge test. Is this a depression-like behavior that can be reversed or prevented by fluoxetine treatment? No. Ketamine promoted the transition from day 1. And as you can see here, is this behavior associated with anhedonia-like behavior? Sucrose tests say no. So now we said, okay, it might not be a depression-like behavior, but this could be a behavioral artifact that is just associated with exposure to electrical shock. So next we asked ourselves, can we create another paradigm, behavioral paradigm, and reproduce this behavioral transition or not? So we teamed up with some colleagues and some engineers from companies, and we designed here a device to expose the animals to inescapable discontinuous swimming sessions. With the device that we have designed, we have a cylinder that is half full of water that connected to a robotic arm, and the robotic arm has a platform at the end where we put the mouse on top of the water on the platform. And then later, the arm would push the platform deep down in the water, leaving the mouse swimming there in the water to choose what kind of action does the mouse want for 15 seconds. After 15 seconds, the mouse is lifted and so on. This process is repeated. We have 3 sessions on 3 different days. And what we notice here, again, we noticed at the beginning that the animals reacted to this discontinuous swimming experience by taking actions. They were swimming in a fierce manner, climbing light, circling in a fast way. And then once the animal realized that there is no escape from the cylinder, we can clearly notice that the animal started to transition to another type of behavior where it's remaining floating in the water. This is the very typical immobile gesture that the mouse shows in water. When we plotted this behavior, we again noticed that we were able to reproduce almost the same pattern of behavioral transition, of this gradual behavioral transition over 3 days exposure of inescapable discontinuous swimming task, similar to what we have observed before with the inescapable electrical shock. Now, is this behavior depression-like behavior that can be reversed by antidepressants? Fluoxetine failed to reverse this one. And again, we did not also observe any anhedonia-like behavior in these mice. So now we moved on to answer a very critical question. Is this a learned helplessness behavior? Do we have a learned helplessness-like status in our mice here? It's very important to separate give-up-like behavior from learned helplessness. With give up, we all know that we try certain tasks and then we fail. Then we just move on and try the next one. And then we might succeed. Then we move on to the next and so on. So the failure in one task is not generalized to all other tasks. We always treat the tasks independent from each other. So whatever your results in the previous one will not really influence, the failure in a previous task will not influence your behavior in the next task. Meanwhile, with learned helplessness, the situation here is quite different. So what happens with learned helplessness is that the subjects are exposed to a major, intensive, uncontrollable, and traumatic event. And here, the subjects will start to develop a perceived feeling of lack of contingency between action and positive output. So they believe whatever I do will not result in any positive output. Therefore, they stop taking any actions when they face any future challenges. So the failure that they have experienced in their previous traumatic event will be generalized. So now is our big question. Would our animals generalize their failure from one task to another, or they will treat these tasks independent from each other? And then when we expose the animals to a discontinuous swimming task first and then tested them in the inescapable shock, or we did the vice versa, we noticed that the animals have treated these tasks independent from each other. The accumulation of failure and the transition of no action in the first task did not influence the behavior in the second one. Next, we did another series of experiments where we combined mathematical computations with behavior. So we actually built a mathematical model in order to construct a curve for the behavioral transition that we are observing in our animals. And we used real data from all the animals that we have tested so far to construct this curve to determine or to estimate the approximate transition point or trial. And we found that most of the animals are transitioning between 55 and 60 trials. And then we prepared 4 groups of animals, exposed them to the IS or the IDS, and then exposed these animals to the classical learned helplessness test in mice, that is the shuttle box test, to see whether or not these animals would exhibit escape deficits like the typical learned helplessness-like behavior in mice. And we tested these animals at 2 different points, and all these experiments were done in independent groups. No group was tested twice. And what we found while we can easily and clearly see the behavioral transition occurred in our mice, the mice did not exhibit behavioral deficits or escape deficits in the shuttle box test, suggesting that the behavioral transition that we are talking about here is not associated with learned helplessness status. Or to be more conservative with our conclusion, at least we can safely say that our behavioral transition occurs way earlier before learned helplessness status can be detected. Now we wanted to move on and use these models to identify the neurobiological substrates that might regulate this behavior. we started first with trying to find which neurotransmitter might be involved in regulating this behavior. I'm not sharing this data with you, but trust me on that. I hope you would believe me. You did a lot of experiments with transgenic animals, with labeling, with dreads to see serotonin and dopamine. We also did behavioral pharmacology experiments and control experiments. And we found that serotonin and dopamine might not regulate this behavior according to our data. Meanwhile, if we activate the noradrenergic system in our animals, we observed increase in the action behavior and suppression of the no action. Now, these animals did not exhibit change in the locomotor activity or in the pain sensitivity. So we concluded here that norepinephrine might be the major neurotransmitter that could regulate this behavior directly without causing any side effects that might influence the behavior indirectly. So we expressed dreads in the lucus ceruleus, and then we activated and inhibited the noradrenergic neurons within the lucus ceruleus, and we saw that activation of these neurons resulted in increase in the action behavior, while inhibition of the noradrenergic neurons, or reducing the activity of noradrenergic neurons, suppressed action and promoted no action in our mice in both paradigms. We moved on to the prefrontal cortex. Which brain region? Human studies basically implicated almost all brain regions in the prefrontal cortex. We wanted to see what is happening in the mouse. And then we started lesioning the prefrontal cortical areas one by one. And we found lesions within the infralimbic, prelimbic, anterior cingulate cortex did not result in any behavioral transition, but lesion within the orbitofrontal cortex actually resulted in suppression of the action, of the no action, and promotion of the action behavior. We expressed dreads in the glutamatergic neurons within the orbitofrontal cortex, and we found, to our surprise, there was no effect on the behavior. But if we express dreads in the GABAergic neurons, we were able to see bidirectional regulation of this behavior. Activating GABAergic neurons in the orbitofrontal cortex promoted action, while inhibition of the GABAergic neurons within the orbitofrontal cortex suppressed action and promoted no action. So next, we did some circuitry and functionality. We combined anterograde, retrograde, mono, and transsynaptic labeling in different Cree lines to define the circuitry and its functionality. Anterograde and retrograde has confirmed that there is intensive projections coming from the LC, from the lucus ceruleus to the orbitofrontal cortex, almost the central part of the orbitofrontal cortex. And when we used retrograde expression, we were able to express dreads in these terminals and in the projecting noradrenergic neurons. And we found, indeed, in line with our previous data, activation or inhibition of these noradrenergic projecting neurons to the orbitofrontal cortex resulted in bidirectional regulation of the behavioral transition, similar to the one observed before. Now we used monosynaptic and transsynaptic expressions and labeling in GAD Cree mice and in KMK2 Cree mice, and we found that majority of the noradrenergic terminals are landing on GABAergic neurons, not on glutamatergic neurons. So now we use FLIPO and DIO to express the dreads in the GABAergic neurons within the orbitofrontal cortex that are receiving projections from noradrenergic neurons. Not in the entire GABAergic population there, only those that receive projections from the LC. And when we activated / inhibited those neurons, we were able again to see bidirectional regulation of this behavioral transition, similar to the one we observed before when we expressed dreads in the GABAergic neurons. Next, we have some neuroanatomical, we have some neurochemical substrates. We wanted to see their dynamic interaction during this behavioral transition. So we use fiber photometry, microdialysis, and mini-2 photon microscopy imaging in freely moving animals. And these experiments resulted in the following findings. We found with fiber photometry that the noradrenergic neurons activity respond to the aversive and controllable trial. Okay, so they start firing at the trial, they increase the activity persistently, and then they decline once the trial is ending. That is the in general. The surprising part, when we look at the overall activity over the time course of the experiment, while we can see clear behavioral transition to no action, we noticed that the noradrenergic activity was increasing. up. That is opposite to what we expected and opposite to our results with dreams. So next we decided to measure norepinephrine in the orbitofrontal cortex. What happened with norepinephrine itself? When we go to the orbitofrontal cortex and we use fiber photometry with expression of norepinephrine sensor in the OFC, or using microdialysis to directly measure norepinephrine within the orbitofrontal cortex, we actually found that over the time course of this experiment, there was a gradual reduction of norepinephrine concentration / probably depletion of norepinephrine within the orbitofrontal cortex that correlated with our gradual behavioral transition from action to no action. Recording from the GABAergic neurons within the OFC gave us very clear image. During the behavioral transition from action to no action, we can see clear reduction in the activity of the GABAergic neurons that correlated with the behavior. And when we carefully analyzed the trials for individual mice, we also noticed that the activity of the GABAergic neurons correlated with the behavioral bouts within the trials, suggests that the GABAergic neurons might be the major drive behind this behavior. Now, to confirm these data and to provide more cellular insight onto the GABAergic neurons within the orbitofrontal cortex, we did imaging, 2-photon microscopy imaging in the orbitofrontal cortex in the GABAergic neurons. And what we found there, first, we were able to reproduce our data with fiber photometry. There was gradual reduction in the GABAergic activity over the time course of the experiment. We also had clear correlation between the activity of the GABAergic neurons and the behavioral choice per trial. But we also found, and another interesting phenomena here, we found that the percentage of the GABAergic neurons that are engaged in this behavior and that are driving this behavior, correlating with what we see at the behavioral level, started to decline over the time course of experiment, in line with the behavioral transition, suggesting that not only the GABAergic neuronal activity is reduced during this behavioral transition, the number, or let's say the percentage of the GABAergic cells that are engaged in driving this behavior also declines. So to summarize and put things all together, I hope I'm not too late. We were able to observe behavioral transition from action to no action when we exposed our animals to prolonged experiences with uncontrollable outcome. Our data suggests that the noradrenergic neurons within the lucus ceruleus projecting to the GABAergic neurons within the orbitofrontal cortex might be among the major regulators of this behavior. What we notice is that the noradrenergic neuronal activity might indicate uncontrollability of the aversive trials that the animal is exposed to, but they do not regulate the behavior per se or the choice of the behavior. Meanwhile, norepinephrine concentration within the orbitofrontal cortex appear to contribute to the regulation of this behavior, probably by controlling the GABAergic neuronal activity. What we notice here that the GABAergic neuronal activity and the percentage of the cells or the number of the cells that are recruited to drive this behavior are important regulator of this behavior. Once the activity declines and the number of the cells that are involved in driving this behavior declines below certain percentage, we start to notice the behavioral transition from action to no action. If you would be just kind and give me a few minutes to speculate and compare these data with other data, because so far I have been stating for you our findings. So if we take this and compare it to what has been reported in the zebrafish, we notice that the mammalian brain seems to be a little bit different. So in the zebrafish, the norepinephrine and the other neuronal and non-neuronal components were activated to inhibit the motor, the futile motor activity. So if we use our language, there, norepinephrine and the activation happens to transition the animal from action to no action. While in the mammalian brain, based on our data so far until now, we notice that the mammalian brain seems to have mechanisms that are driving the no action by default. Meanwhile, we have other mechanisms like norepinephrine and GABA. They try to delay or prevent this transition to no action as much as possible. And once these mechanisms are depleted, the behavioral transition occurs. At the end, I would like to thank all members of my lab. Most of the work that I have presented today is done by my postdoc, Li Chaochun, by my PhD student, Sun Tianping, and my associated professor, Dr. Li Wei. This work has been supported by the NSFC grant, and I would like also to acknowledge the kind and very, very generous help from our colleagues here in China, my colleagues here in China, Professor Rawi for the transgenic animals and the knockout TPH2 knockout animals, Lo Mingming for the Cree lines, Zhu Zilong for the Dreeds clones and vectors, He Miao and Zhang Jiayi for the Cree mice. You probably can see that we have used a lot of Cree mice and different lines of Cree mice in this study. Our collaborators, Chen Haping, Yuan Shi, he's an academy member. Professor Chen Haping is an academy member. Zhao Ting from Peking University and Nanjing Imaging Center for their kind help and support with the 2-photon microscopy imaging and data analysis. Their support was so great and generous. I also would like to acknowledge Kaivan and Gu Yu. Those are long-term collaborators with my lab. Finally, for you all, I'd like also to say that most of the images and the beautiful pictures that I use here were done and designed by this lovely and talented young lady, Zhu Anghui, here in Shanghai. And thank you all for your attention. Thank you.
Great talk, Nash. And we have time for questions.
Nash, is Yi here?
Yeah.
It's a great talk. It's really, really interesting.
Really? Thank you. Thank you.
Thanks. Yeah. So can you comment on how does the OFC link to the motor system?
Yeah. Yes. I'm so here. I can comment, but I would please consider that speculations. Actually, we are working on this. It's not an easy task. As you can see, we started from scratch here, and we are collecting the pieces of the puzzle on our way, one by one. So here, when we reach this point, and currently we are at a situation where we don't know anything, there are so many possibilities, and we are collecting the pieces. We do believe that there must be somewhere another area where those GABAergic neurons are causing disinhibition. So those GABAergic neurons are expected to cause disinhibition somewhere else. However, the surprising part that this area, until now at least, doesn't seem to be in the other prefrontal cortical areas. But we are still searching. So we have possibilities it might be another brain area deep in the brain, backside. So I would say so far, what I'm allowed to say, there might be, those GABAergic neurons will be projecting somewhere else, and they are causing disinhibition in another brain area that has intensive glutamatergic neurons to control this motor behavior.
Thanks.
No, thank you so much. Thanks for the kind comment.
Hey Nash, this is Chen here. It's a wonderful talk. I'm curious if you can speculate on the difference of the generalization between IS and IDS. Like why in the same context the animal would learn to give up at the in a different context the animal would learn to keep struggling, and what would be the original signal from? Does the locus coeruleus decide, or does the locus coeruleus receive inputs from, for example, MPFC, like the like dorsal raphe nucleus?
Okay. It's Ren Chen, right? Am I right? I recognize the voice. Okay, so you need to be slow with me. My brain is not as smart as yours. You are too fast with so many questions. So I would start with the beginning. So you have a very beautiful point. You say that here perhaps the change in the context might have resulted in prevention of the generalization, right? So it might result in the observed separation between the tasks. Well, that would be in contrast with what has been repeatedly shown before by Meyer. You know, the learned helplessness is actually generated by Seligman and Meyer. That's the original literature. Actually, Meyer shows that when the animals are exposed to shock in one context, moving them to another one for the escape test was not influenced. The generalization happened, and the animals did show escape deficits. Now, the contextual contribution to this kind of behavior, I think, has been excluded by some lesions in different areas, and including amygdala. And they notice that the contextual element here does not really have a major effect. So you need to go back to Meyer literature on this part, I think in the 90s, during the 90s. That's the first part. Second part is, yes, again, the situation here is endless number of possibilities and questions. We did provide a snapshot on something in the middle. Who drives the behavior in the lucus coeruleus on the first place? I can speculate, I can give you possibilities now, but I cannot give you a definite answer. I would not be able even to give you a most likely answer, because it could be some sensors from the brain stem. It could be from simply from the peripheral nervous system moving up to the brain stem and driving the noradrenergic projections to the prefrontal cortex. It could be a feedback from the prefrontal cortex to the noradrenergic neurons. There are multiple possibilities. It could be another brain area within the midbrain that controls the firing activity of the noradrenergic neurons. These are all many questions that needs to be answered. What I'm trying to say here, I'm trying to say we just did the first step. Do you understand me? You need to give me some time. We can't really cover the whole area once in one study.
Oh, thank you. Beautiful first step. Yeah.
Okay, Nash, as always, awesome, thought-provoking talk.
Thank you so much. Thank you.
Okay.
So I will stop sharing now, right?
Right. And Zalong, you can introduce Norbert.
Zalong can go ahead.
Thanks, that's a great talk. Now I have a second speaker from Indiana University, Professor Norbert Harris. So actually, Norbert is grown up in Hungary as has academic training and as well as the professional experience in Hungary, and after getting a PhD and also as I believe just something as a doctor of sciences also in Hungarian Budapest. One of my favorites, the city. I haven't been visit there. So Norbert has started his professional lab in Hungarian study. Particularly neurocircuitry underlying the interneuronal circuitry in the brain associated with a variety of physiological activities from last year, 2021. So Norbert become joint professor in Indiana University as well as the Institute of Experimental Medicine, where he started over a decade. So today we're very happy to have Norbert to give us the talk to talk about the latest about the midbrain amygdala pathway, also underlying interneural activities to control the defensive behavior. Welcome, Norbert. Okay.
Thank you. Thank you so much, Zidon, for the kind of introduction. And I try to share my screen, and I hope that you can see my screen now.
Great.
Yeah. Perfect. Thanks so much. Today, I like to introduce you a hitherto unstudied pathway originating in the midbrain and which terminates in the amygdala and play a role in controlling the defensive behavior. So, irrespective of the size of the animal, they may face with life-threatening challenges during the course of their life. For instance, they may become a prey in a given situation, as you may see on these images. So earlier, it has been noticed that actually the primary factor which determines the behavior of the prey is the distance between the prey and the predator. So if the distance is between the prey and the predator is large enough so the animal can escape, there's a possibility for the escape, then the prey will swim, run, or fly away. However, there is no escape, there is no possible, then the prey will freeze, hoping to be unnoticed and avoid attack. But if the predator and the prey get in a close vicinity, then as a last chance, the prey will attack the predator. This defensive attack is closely related actually to the panic behavior, as it contains many unpredictable movements with the aim to confuse the predator as much as possible and find a chance to escape. Does the prey even meet a predator? There's three different behavior outcomes: either flight, or freeze, or fight. So what are the neuronal circuit underlying the defensive behavior? So the center of the defensive behavior in the brain is actually the para-aqueductal gray, as the neurons in this region control directly the different motor circuit, activation of which actually leads to distinct defensive behavior. An appropriate defensive reaction aiming to avoid predator attack relies on sensory information, which originating from the tactum or to the parabrachial nucleus or the spinal midbrain tract. And PAG, the para-aqueductal gray, is not simple reflex center at all, but is operating under the control of tightly controlled by higher order centers, for instance, like the extended amygdala, which has two parts, the bed nucleus terminalis and the central amygdala, as well as by hypothalamus. And this region which directs the PAG neuronal circuits to select the best behavior outcome under the given situation. And on the top of the hierarchy, there is cortical structures that control the function of the extended amygdala as well as the hypothalamus. But you may actually notice that the appropriate function of such a system is need a feedback information that originate from the downstream region, and that would help to precisely control the outcome of at the behavior level. So this operational principle allows rapid and very effective adjustment if needed upon external challenges. So we know that the extended amygdala actually adjusts the cortical regions indirectly via the cholinergic neurons sitting in the basal forebrain. But what about the PAG? So do we have feedback input from the PAG, from the para-aqueductal gray towards the extended amygdala? So if yes, what is the role of this feedback projection which controlling a defensive behavior? So these are the questions which I try to address in my present talk. So in the past years, we have begun to study a neural population in the para-aqueductal gray, and these neurons are actually express vasoactive intestinal polypeptide, and you may see here shown in green that they are situated in primary these neurons in the ventral para-aqueductal gray, and small portion of them actually extend to the dorsal raphe. So if we inject a viral vector, a tracer, into the VIP mouse, selectively label these neurons, we are very much surprised to see that the axonal projection from the VIP neurons from the PAG actually selectively present in the BNST, in the bed nucleus terminalis, in both sides, as well as the central amygdala. So these neurons, these VIP neurons in the PAG, selectively innervate only the extended amygdala. We can't see any axons in the entire brain. So there is a question then, what we ask first: do these VIP neurons in the PAG convey information to the different parts of the extended amygdala, to the BNST and the central amygdala, separately or simultaneously? So there is a separate neuronal population which project either to the BNST or to the central amygdala, or the same neurons may project ipsilaterally to the BNST and central amygdala, or there's a third possibility whether the neurons project simultaneously to the entire extended amygdala region. So to address this question, we used the following strategy. We injected a viral vector into the BNST on one side, and we waited 3 months. This long time allowed us to label retrogradely the neuronal population at the PAG level. You can see the cell bodies here, as well as we can label the axon collaterals of these neurons, and what we in an anterograde manner. What we have seen after injection to one side of the BNST, that we could see external labeling shown here in yellow in the central amygdala ipsilaterally, as well as contralaterally, as well as we can see axons in the BNST in the contralaterally. Thus, what we can conclude, the neurons in the VIP can broadcast simultaneously the information to the BNST and to the central amygdala into the both hemispheres. So the next question we asked, what is the input? What is the monosynaptic input to this VIP neurons sitting in the PAG? So therefore, we use a monosynaptic rabies tracing. And so we injected these viral vectors into the VIP mice PAG region. And we look for the neurons which are monosynaptic innervate these VIP neurons. What we observed that the majority of neurons, more than 50%, are actually, which innervate the VIP cells, are present in the BNST and the central amygdala. So those regions which get innervation to VIP neurons, they actually project there and they innervate the VIP neurons. Okay, so there's a feedback loop between the extended amygdala and the PAG in this respect. In addition, we could see neurons labeled in the hypothalamus as well as the parabrachial nucleus and the other regions in the brain. But the majority of the neurons, what we could observe with this technique, was were present in the BNS in the central amygdala. So what is the output of the neurons? This is what we have asked next. To do that, we use optogenetics. We injected into the VIP mice vector, which allowed us to express channel rhodopsin in the exon terminals of these fibers. And then we made slice recording, both in the BNST and the separate experiment to the central amygdala. And we evoked transmitter release from the exon terminals using the blue light stimulation. And you can see there is a two example here. One neuron was recorded in the BNST, the second was in the central amygdala. But you might see that upon 5 consecutive blue light stimulation, there's an inward current, what we can see, what we can record in the neurons, in the postsynaptic neurons in the BNST on the central amygdala. And this inward current was fully blocked in both cases with the ionotropic glutamate receptor blocker, the MBQX and AP5. And we can wash out partially this antagonist effect. So what we can conclude, that the glutamate is released from the exon terminals of the VIP neurons in the target region in the extended amygdala. Next, we make electron microscopy. Next, we actually try to find out what are the postsynaptic targets of these neurons, of the VIP neurons at the level of extended amygdala. We included biocytin in the recording pipette, okay, and we labeled the neurons in the central amygdala and in the BNST. And we, as you may know, in these 2 regions, there is two major GABAergic cell type. One express somatostatin, the other express PKC delta, the protein kinase C subtype delta. So what we did, we fluorescently labeled the recorded neurons, and then we made multiple fluorescent staining. And what we were surprised that the majority of neurons which received excitatory input from the PAG via this VIP projection didn't contain PKC delta or somatostatin. The only 1/3 of the total population had we could observe, visualized with fluorescent immunized staining, PKC delta, and a small portion of the postsynaptic target did express somatostatin, shown on this example. So what we can conclude, the majority of the postsynaptic target of these VIP fibers at the PAG level are actually neither PKC delta cells nor somatostatin cells. So at present, we are looking for this cell population, which might be primary target of these VIP afferents at the level of the extended amygdala. So next, we wanted to unravel what is the subcellular target of the VIP afferents at the level of the extended amygdala. Therefore, we made electron microscopy, and we look for the postsynaptic targets of the VIP expressing exon terminals, both in the central amygdala and the BNST. What we found that the majority, more than 75% of the total, the targets are actually spines. And that only a small portion, 1/4 of the portion is a small caliber dendrites. And the exon terminals form, the VIP exon terminals form asymmetric synapses, which are, again, consist of the idea that these pathways actually excitatory, so they release a glutamate and they activate the postsynaptic targets via ionotropic glutamate receptors. From the previously studies, we knew that VIP neurons, at least portion of them, may express dopamine. Therefore, we made a dual immunostaining. So we revealed the VIP content of the neurons at the PAG level, as well as we made immunolabeling against the tyrosine hydroxylase, which is a rate-limiting enzyme of the synthesis of dopamine. As you may see here, that a portion of the VIP neurons indeed were immunolabeled for the TH, so the tyrosine hydroxylase, i.e. they are dopaminergic. We found that 25% of the VIP neurons using that immunostaining were verified that they are dopaminergic both in male and female as well. To get kind of a support for this data that these excitatory glutamatergic cells at the PAG level may be actually dopaminergic, and maybe a larger portion of them, but we can't see with the immunolabeling. We injected 6-hydroxydopamine into the PAG region, and we counted this loss in a way. And we noticed that there is a large change in the number of those cells which express only VIP or express TH, i.e. These VIP neurons are very likely, the majority of them are actually dopaminergic, but with the immunohistochemistry, maybe the level of the TH is below the detection threshold of using our methods. So what this first part of the presentation, which I just like to summarize, that we have identified a neural population in the ventral para-aqueductal gray, which express VIP, as very likely a large portion of them are actually dopaminergic. They release glutamate at the target regions, which are the extended amygdala, and they receive the major input on these VIP neurons are actually originated from the extended amygdala, from the BNST and from the central amygdala. So the next question, what we asked, what is the function of these neurons in the defensive behavior? So therefore, we used two strategies. First strategy was to stop the neurotransmitter release from the exon terminals of the VIP neurons. We express them in a Cre-dependent manner, tetanus toxin light chain, which is inhibit the synaptic vesicle docking in the exon terminals by proteolytic cleaving a synaptobrevin, i.e. the neurons are intact, we have a dendrized axon, yet the neurotransmitter is not released from the exon terminals. We had two groups of mice. There's a control group which express only the fluorescence protein, whereas the other group express this tetrodotoxin light chain, which mute basically the VIP neurons. So we first we assess the defensive behavior using Pavlovian field conditioning. We use this classical method where in a box in context A we present the tone, and which is co-terminated with the electrical shock 7 times, and the next day we test for the contextual field memory of the both group of the mice, and then in the following days we tested in a different context, in context B, the acute field memory as well as the extensional learning. What we have observed in compare the control and the TELS group, the tetrodotoxin light chain expressing group, there was no change in the field memory acquisition during the tone presentation and the presentation of the co-termination of the shock. But we see, what we have seen, significant difference in the contextual field memory and the second day. Those mice which express tetrodotoxin light chain in VIP neurons, they showed less freezing, significantly less freezing compared to the controls. But we couldn't see any difference in the acute field memory, neither in the nor in the extensional learning. So what we have observed that the contextual fear memory was selectively impaired under our conditions, and the acute fear memory and the extinction area was not changed. Next, what we have done, we assessed the predatory odor avoidance test, whether these VIP neurons may play a role in this task. So we put the mice into an arena. In one corner of the arena, we present a potent analog to the predatory odor of this trimethylthiazine, which is a main component of the fox and cat and other predator visit. And we placed a small amount of this 2-MT, the modified odor analog, to 1/4. And then we just assess the approach and avoidance behavior in these mice. But we have observed that in the group of these mice which express tetanus-like change in the VIP neurons, there was a significantly less freezing. They spend less time in the avoidance zone, as well as they more often approach the zone where the TMT was present in the corner. What we can conclude that the VIP neurons actually control the predatory odor avoidance as well, in addition to the contextual fear memory. Next, what we have assessed that how these VIP neurons play a role in contextual fear acquisition. So therefore, we use a chromatogenetic approach when actually we express an inhibitory dread in the VIP neurons, as well as the other group of neurons, we express a control virus. And we made the very same experiment, except here with 30 minutes prior to the contextual fear learning, we injected both group of mice with the CNO, which is the clozapine nitric oxide, which is a selective agonist of the dread receptor. And then we made the very same protocol as I just described previously. And what we have observed, there was no difference in the fear acquisition in the both group of mice. So the control and the dread inhibitor dread expressing mice both received the CNO injections 30 minutes prior to the acquisition, showed no difference. Then we wait 2 days just to be sure the CNO is gone from the animals, and we placed back to the very same context in mice. And again, we have seen significant less freezing in the context A in mice which express inhibitory dread compared to the controls. It means that the fear acquisition, actually during the fear acquisition, VIP cells sitting in the PAG play a role. But again, consistent with the test data, there was no change in the cued fear memory as well as extinction learning in the following days assessed in a different context. And the last, we assessed the effect of these VIP cells in the predatory odor avoidance. Again, we made the very same protocol, but with the 2 groups of mice, we have seen 30 minutes before the testing. And we have seen that the inhibitor dread expressive mice enter more often to the odor zone, as well as they spend less time in the avoidance zone and made more often, they approach more often the odor zone where the predatory odor was presented. So to sum up my talk, I would like to just, so we found that identified the neurons in the ventral pug which express vasoactive intercellular polypeptide. A portion of them are dopaminergic cells, and they can release glutamate in the postsynaptic target in the bad nucleostrial terminals in the central amygdala. And the inhibition of the function of these pug neurons expressing VIP results in an impairment in contextual fear learning and predatory odor avoidance, a set of defensive behavior. So what we have found, we have identified a projection, very likely a feedback projection from the pug, which innervates specifically the extended amygdala and play a role in defensive behavior. And last but not least, I like to thank my colleagues in the Budapest laboratory. Primary Kinga and Laura was the primary driver of this experiment with the help of Zsuzsi and Orsi. And I like to also thank my collaborators in Europe who helped me to figure out, to help us to figure out this function of this one. And thanks so much for your attention.
Great, thanks, Norbert. Great talk. Now I open for questions. I start the first one? Yeah, I'm gonna start the first one. So I'm curious about regarding to the separate output. Well, would you be suspecting that there be maybe different regarding if you retrospectively synaptically tracing, like putting AV retrograde to the either BNST or say the central amygdala? Would you be expecting the two different output play different role in kind of offensive behavior?
I would expect the different. Actually, I would expect that this VIP pathway would control the very same type of behavior.
Okay.
I don't expect that they would control different behavior. We have very interestingly, we have injected a retrograde tracers to the BNST as well as to central amygdala. And what we have seen that the very same regions project simultaneously to these two parts of the extended amygdala. Based on this connectivity, at least I don't expect that these VIP cells and the extended amygdala and these two parts of the extended amygdala play a different role in defensive behavior. I think they work together in that set of behavior outcome, and I don't think so that they play a different role.
Okay. But if physiologically, I believe maybe I didn't follow closely about the role of BNST kinds of behavior before. So yeah, I mean maybe some other behavior, right? By the projecting different target in the brain. So I suspecting where be something different.
I think let put this way. I'm not saying that BNST and extended amygdala may at all time they do the same job, which I think that the postsynaptic targets of this VIP afferent, which is at present unknown. Okay, this is a different cell population should be, since they don't express somatostatin or the PKC delta, so there might be a similar, there's maybe a certain population, and that population either present or extended amygdala or in the central amygdala may have the same function. Yet these somatostatin cells and PKC delta sitting in the extended in the BNST or the central amygdala, they may have been recruited during different tasks and during different conditions. Okay, this is my model which I'm thinking of at present.
Great, thanks. Any more questions? I'm curious, do you have any molecular marker for the unidentified cell population yet, by the way?
Not yet. We are working on it hard to find out.
Maybe use some single cell sequencing in the future to figure it out.
That's correct. That's the direction what we are actually trying to use. But at present, I can't really share any data because we haven't really picked up the cell type. That would be a really goal.
Can I ask a question?
Sure. Go ahead, Nash.
Would that be possible? Norbert, thank you for this nice talk. That's beautiful. May I ask you a question? It might be I might have missed some detail. You might have said something about it because I was answering some questions at the beginning, or it could be also my question could be a sort of overinterpretations. Have you tried to see whether or not there are some connections between these projections and the visual system to detect the danger, the location of the danger, the direction of the danger, whether it's coming towards you, whether it's going away from you? Are you having such plans to do that on these?
There is no, based on the rabies tracing experiment, we don't expect to see a direct visual input on the VIP cells sitting in the park. They can get indirect response. I'm not against it. Actually, we are planning to test this possibility in looming using a looming stimulation.
Exactly.
But we haven't done yet. But based on the connectivity, I don't expect a major change. But we have to make an experiment, and afterwards
yes, because because the revealing like this aspect and checking out with the looming would be very helpful. And that would be very beautiful if you can differentiate those three. You remember your first slide of the different responses to the danger. That would be great, actually. Yeah. Thank you. Thank you.
Thank you, Nash. Thank you.
Okay, more questions. Thank Nash and Norbert for great talks today. Thank you.
Thanks so much.
See you all next week.
Thank you, guys. Thank you.
Thank you. Thank you so much for
everyone.
listening. And invitation. Bye-bye.
Bye-bye.
Thanks, everyone. Thanks, Nash. Thanks, Norbert.
Thanks, Aaron. Bye-bye.
Thank you, Aaron.